All right, gentlemen, let's jump right into chapter 11 today and talk about Mendel and the gene idea. We've talked about this a little bit in class, and I'm going to go through it in this slide deck. And remember that a lot of the material for genetics, a lot of the material we're doing in chapter 11 is trying to help you with vocabulary, making sure that you have language to talk about the things that we're going to discuss in this class. There is some math involved, some things involved that you'll find that it's probably best to just try working on problems, but hopefully by going through these slides, you'll be able to know some of the basic ideas that Gregor Mendel helped to develop and some of the ways we apply those to our understanding of genetics. So Mendel is a German monk who studied pea plants, and you've probably experienced people talking about these pea plants in science classes before. There were purple flowers and white flowers, and yellow peas and green peas and tall and short, all these different traits that Mendel was able to study. So the first part of chapter 11 talks about how Mendel used the scientific approach to identify two laws of inheritance. These two laws of inheritance should be relatively straightforward for you to understand, especially since we're coming right off of meiosis, but nonetheless we'll talk through how Mendel sort of arrived at them. Now, remember that Mendel did all of his work without having an understanding of the genetics or the molecular biology of DNA, anything like that. So, he talked about things such as characters in his plants. These were varieties that had certain traits. So a character might be flower color, a trait would be purple or white. And so he sort of identified these characteristics, these traits of plants, and then he could control the mating between his plants, and that's why he used pea plants in particular for the work that we still talk about. Mendel actually studied a handful of other plants. Not all of them were as clear-cut as pea plants, and so they didn't allow an easy explanation of the laws of inheritance. So as you can see here, when you think about how he would do these experiments, the pea plants have their sex organs sort of inside the flower. He could clip them off with a pair of scissors and then use a paintbrush to specifically pollinate one type of flower with the pollen from another type of flower. Therefore, he would know specifically who the parents of a plant were and could then look at those offspring and try to figure out what their traits were. So you already see right here some of the language, P for parental generation, and then the F1 generation is the first filial generation. They're the offspring of those parents. So Mendel did some early experiments where he took true breeding white and purple flower plants. These are plants that over time you would breed them with themselves. So a white plant bred with itself again. For generation after generation would only make white plants. Purple plants bred with themselves generation after generation would only make purple plants. Mendel called them true breeding. They would only make the type of plant that they were. He then took, like a good scientist, the white and the purple true breeds and mixed them together. And all of the F1 plants that he got out of that were purple. And I'll show you a picture of this in a second. So all of the F1s were purple. He took those F1s and he crossed them with each other to make an F2 generation, a filial 2 generation. And then discovered that he had a ratio of 3 to 1 purple to white. So the white flowers reappeared after mating the F1 hybrid flowers from his original true breeding parents. So true breeding purple, true breeding white, put them together, everybody's purple, take these purple flowers and mate them, and you get 3/4 purple, 1/4 white. Pretty consistently he would see this pattern. And so what Mendel took from this, what he understood from this, was that the white characteristic, the white factor, as he called it, was hidden or masked by the purple factor. And we now describe these as dominant and recessive. So a trait that covers up another trait is called a dominant trait. Purple was dominant. A trait that is covered up by a dominant trait is called a recessive trait. So white is a recessive trait. Importantly, Mendel, and we can observe that the white flower trait does not go away. And we know this because it comes back. So even though that we don't see white flowers in the F1 generation, the fact that white flowers reappear in the F2 generation shows that the traits for white flowers, the heritable factor for white flowers, doesn't go away. It's just hidden in that F1 generation. Now, like I said, Mendel didn't know anything about molecular biology, but you all do, and so you know that the heritable factor that we're talking about here is a gene. So the gene for white is recessive, but it doesn't go away. It's not diluted or destroyed through this process. So as he studied these pea plants, Mendel saw these different characters, and the same thing reoccurred multiple times. So between purple and white, purple is dominant to white. The F1 generation would all be purple. The F2 generation would show you a 3-to-1 ratio of purple to white. Same with seed color, yellow dominant to green, round dominant to wrinkled, inflated dominant to constricted, green dominant to yellow, axial dominant to terminal, tall dominant to dwarf, always getting a ratio around 3-to-1. As I mentioned in class the other day, there is some data that suggests that Mendel may have manipulated his numbers a little bit to get data that looks this good. It's possibly statistically unlikely to get a perfect ratio on all of these characteristics and all these experiments, but his underlying conclusions are not incorrect. It just suggests that maybe some things that didn't work the way he expected to, he just threw out. So from these observations, he developed a model, and this model hopefully makes sense to you, but let's talk through it so that we can think about all the parts. So there were 4 parts to this model. One was that alternative versions of a gene account for variations in inherited characters. I'm putting gene in here, but Mendel just called this a heritable factor. So there were alternative versions. The word we'll use for those alternative versions are alleles. And so you have an allele for purple color and you have an allele for white color. For each character, an organism inherits 2 alleles. So you get 2 alleles. You could have purple-purple, you could have white-white, you could have purple-white. If they're identical, purple-purple or white-white, we would call that a true breeding organism. That's the only alleles that it has, white or purple. If the 2 alleles at the locus are different, purple-white or white-purple, which coincidentally are the same thing, then one, the dominant allele, determines the appearance, and the recessive allele has no noticeable effects. So if you are purple and white, your flower appears to be purple, not white. And these 2 alleles for heritable characters separate during gamete formation. So that whole last chapter on meiosis basically set the stage for you to recognize that these characters separate during gamete formation and end up in separate gametes. This is actually the law of segregation. Purple and white will go their opposite directions and they'll end up in 2 different gametes. You can see this, again, the same picture we saw before, but now we're going to show these genes, these alleles. So we're going to use, by convention, capital letters to represent dominant traits and lowercase letters to represent recessive. So a true breeding purple flower would be represented by a capital P, while a true breeding white flower would be represented by a lowercase p. When these cross, when we make an F1 generation, the only gamete that the purple flowers can give is a big P. The only gamete that the white flowers can give is a small p, and so all of the F1 generation have a big P and a little p. They are purple, but they have both genes inside their genome. When they make gametes, half of their gametes will be big P, and half of their gametes will be little p because of the process of meiosis, which means that when we combine one of these purples with another one of these purples, we can make this structure, this diagram called a Punnett square, where we put that of the 2 possibilities of a sperm, one is a big P, one is a little p. Of the 2 possibilities of an egg, one is a big P, one is a little p. And then by combining the letters, we get the 4 possible outcomes from the sperm and egg. So we can get a big P, we can get a big P, little p, we can get a big P, little p again, or we can get a little p, little p. All of the ones that have at least one big P will be purple, all of the ones that have no big P will be white, and so we end up with this 3-to-1 ratio. Punnett squares are a tool that are really helpful for allowing you to do quick probabilities. And we'll use them in this class, but I'll also show you some of the ways to just do the math yourself. And eventually you won't need the Punnett square to be able to see what's happening in a hybridization like this. So I've used some of this vocabulary already, but let's just go through it to make sure that you have it all down. If you have two identical alleles for a character, we're going to call that homozygous. Homo for same, zygous referring to the zygote, so big P, or little p is homozygous. We can describe it as dominant, homozygous dominant if it's big P, or homozygous recessive if it's little p. If it has two different alleles, we're going to call it heterozygous. Heterozygous, hetero for different, two difference in the zygote. That would be a big P or a little p big P, which, as I've already mentioned, we're not going to treat big P any different from little p big P. It's still a mixture of two alleles. Heterozygotes would not be true breeding, but homozygotes would be. As we think about these descriptions of genes, the genotype is going to be talking about which alleles an organism has, while the phenotype will describe its physical appearance, what it looks like. So you can have two different genotypes, homozygous dominant or heterozygous, that gives you a purple flower, but there's only one phenotype there, and that's purple flower. So phenotype is going to describe what you see, genotype is going to describe the alleles. And you can see this here. Homozygous dominant, big P, is going to be purple. Heterozygous is also going to be purple. Homozygous recessive is going to be white. So you have a 3-to-1 ratio of phenotypes, but that actually represents a 1-to-2-to-1 ratio of genotypes. So as you think about these probabilities, one of the tools in a geneticist's toolbox, one of the tools in Mendel's toolbox, was that if you have an individual with an unknown phenotype, you can figure out its phenotype by crossing it, mating it with organisms of known genotypes, and then infer from the results what's going on. So if you have a dominant phenotype flower, in this case you have a purple flower, and you're not sure whether it's homozygous dominant or heterozygous, you can do a test cross, breed it with a homozygous recessive individual, and try to figure out what's going on. So this is simply a puzzle, and I think what guys like about genetics, what they like about Punnett squares, is these are relatively straightforward problems to solve. So you have an unknown, you don't know whether it's big P or big P. You cross it with a known. You always are going to test cross with a recessive phenotype, because recessives are the only things that you can actually know that they're little p, they're homozygous recessive. But if this dominant phenotype flower is homozygous dominant, you would expect a big P crossing with a little p to give you 100% big P, all of the offspring will be purple. On the other hand, if this unknown flower is heterozygous, a big P crossing with a little p should give you 50% purple and 50% white, and that would let you know what the dominant phenotype flower's genotype was, whether it was heterozygous if you get this result, or homozygous if you get this result. I know I'm going through that really quickly here, but that's definitely something that you can get a piece of paper, write it down, make sure that it makes sense to you. So aside from the law of segregation, the second law that Mendel discovered was the law of independent assortment. Now, so we talk about peas as if, purple and white flowers are the only thing going on, or yellow and green peas are the only thing that's going on. But we know that plants with flowers also have peas. And so Mendel started looking at more than one character at a time. So he started looking at, flower, pea color, and pea shape, or things like that. And so he did what was called a dihybrid cross. So if we're only looking at one character, we're going to call it a monohybrid cross. If we want to look at two characters at the same time, we're going to call it a dihybrid cross. Technically, you could do a trihybrid, a tetrahybrid, a polyhybrid cross. The bigger these crosses get, the more complex the math is going to be. So we're not going to do those in this class, but recognize that what I'm showing you could go up, forward to as many characters as you want. So now we're going to look at peas. So yellow and smooth peas are homozygous dominant. Yellow and wrinkled peas are homozygous recessive. So when he crosses those together, he gets an F1 generation that is all yellow and all smooth, because those are the dominant traits. Now, there are two ways that this could have happened when he gets now the F2 generation. If you saw a simple 3-to-1 ratio, then you would know that yellow and wrinkly traits go with each other. So you can't be yellow and wrinkly, you can only be yellow and smooth, you can only be green and wrinkly, and that's the only thing you would see. That would suggest that these characters sort with each other. They're dependent on each other for their sorting. On the other hand, the hypothesis of independent assortment, which is actually the one that we're looking at here, shows that yellow and green color is separate from wrinkled and smooth, and so you can get offspring that have yellow color but wrinkly traits. You can get offspring that have green color but are smooth, and you end up with a phenotypic ratio of 9:3 to 3:1 in this type of problem. Now, fundamentally, and we'll look at this in class, what's going on here is that because the alleles are all sorting independently, you have a 50% chance of getting a big Y or a little y, and you have a 50% chance of getting a big R or a little r. And so we have to combine those probabilities. So the chance of getting a big Y and a big R is 50% times 50%, or a quarter. A big Y and a little r is a quarter, a quarter, a quarter, and then we work these probabilities out as we go forward. This is a difficult thing to do. I'll probably make another video where I'll show you how to do a dihybrid cross, but hopefully if you look at it here, this makes sense that that these traits are sorted separately from each other. So it doesn't matter whether you're yellow or green with regards to whether you're wrinkled or smooth. So the law of independent assortment specifically says that any pair of alleles segregates independently of any other pair during gamete formation. Again, if you think about meiosis, this should make sense. The challenge is that this is not always true. Just hang on to that idea for right now. So genes that are near each other or on the same chromosome can be inherited together. Mendel never looked at this or never recorded this. We will actually talk about that in the next chapter. So the law of independent assortment is true unless there are other mitigating factors, and hang on to that for later. It makes things way more interesting. Alright, so that was concept 11.1. 11.2 is really quick. It's basically a math review, and I've kind of already done it for you. Probability laws are what's doing all of this Mendelian inheritance. We often think about which allele goes where as a coin flip. So multiplication rule states the probability of two or more independent events will occur together is the product of their probabilities. So what is the chance of getting a heads followed by a tails is going to be 1/2 times 1/2. You're going to get a heads followed by a tails a quarter of the time. The addition rule states that the probability of any one or two mutually exclusive events will occur is by adding together the probabilities. So in this case, what is the probability of getting both a heads and a tails if I flip a coin twice? In that question, you don't care which order. So the probability of getting a heads followed by a tails is 1/4. The probability of getting a tails followed by a heads is 1/4. So what is the probability of getting a heads and a tails together is going to be 1/4 plus 1/4, or a half. Hopefully when you look at the Punnett squares, this kind of math makes sense to you, but we can definitely go over it if it seems a little confusing. And there are some nice pictures here, and this is how you get half, and a quarter is telling you what the probabilities of each event are. And when we talked about genotypes and we said 1 to 2 to 1, we really think we're going to operate as if little r, big R is the same as big R, little r. Heads and tails is the same as tails and heads. So we really will add these two together. So you get a 1/2 chance of getting one of these possibilities, a quarter of this, 1 to 2 to 1. You can use these to predict the outcomes of crosses. So the Punnett square are a shortcut way to see this, but you can always just determine the probability of each separate gamete and then multiply them together. So if you have big Y, big R, little r, those dihybrid crosses, the probability of getting a big Y, little r is just the combined multiplied probabilities of each individual event. So a quarter for big Y, a quarter for big R. 1 out of 16 will be completely homozygous dominant. If we want to know what's the probability of getting big Y, little y, heterozygous for Y, and dominant, homozygous dominant for R, we now are a half times a quarter, 1 out of 8. You can do this all together for that whole Punnett square, and you'll see that the probability of, this is for 3 traits, right? And so this is where it gets really complicated, but you can see the chance of at least 2 recessive traits can be calculated by pulling out the separate pieces here. These are, again, things you'll have to play with, and we'll work on to make sure they make sense to you. Alright, so now where we start to get interesting, right? Mendel used some very simple ideas to think about inheritance patterns when he talked about yellow and green or purple and white. And as it turns out, the more you look, the more complex genetics gets. If you've been in my class all year, you shouldn't be surprised that the more we look, the more complex biology gets. And so not all heritable traits, characters are as simple as the ones Mendel studied. The basic principles of segregation and independent assortment will still apply. Genotypes will still be created in the way that genotypes should be created because you understand how the molecular basis for that works. But sometimes phenotypes are going to look different depending on what's actually happening. So the 3 situations we're going to talk about here is that sometimes alleles are not completely dominant or recessive, so you get more of a mixture than getting one or the other. Sometimes a gene has more than 2 alleles, so if instead of purple and white we talk purple, red, and white, you might get some different combinations. And sometimes a single gene produces multiple phenotypes, and so there's more things happening than one simple thing from one simple gene. Let's talk about degrees of dominance first. Complete dominance is what we already have talked about. The phenotypes of the heterozygote and the dominant homozygote are identical. Other 2 degrees of dominance are incomplete dominance and codominance, and in both of these cases the heterozygotes are going to look different. In incomplete dominance, the heterozygotes are somewhere between the parental varieties. So if you have red flowers and white flowers, the heterozygotes in incomplete dominance will be pink. You're going to see a mixture of red and white. In codominance, the 2 dominant alleles affect the phenotype in separate but distinguishable ways. So again, the heterozygotes are going to be different, but the heterozygotes between a red and white in codominance are going to be red and white flowers, maybe spots or speckles or stripes or whatever. If you get down to the tiniest molecular level, incomplete dominance is really codominance at a microscopic level. So I really treat incomplete and codominance as very similar concepts. But definitionally, incomplete dominance, the heterozygote is going to look like a mixture. Codominance, the heterozygote is going to look like both of the parents. So here's an example of incomplete. Red and white make pink. And I don't have an example of a complete a codominance, but red and white would make red and white. Alright, multiple alleles are also a really interesting situation here. So multiple alleles are when you have more than 2 forms of the gene. The 4 phenotypes of blood that we see in humans are determined by 3 alleles for blood type, I for isotype. So you can have an A isotype, you can have a B isotype, or you can have a recessive isotype that is neither A nor B. This notation is kind of interesting. So the I is showing you that the isotype is dominant. The A is showing you that it's type A and type B. So A and B are both dominant over I, but A and B are not dominant over each other. They are actually codominant to each other. So when we see this happening, IA or IAI are going to give you type A blood. IB or IBI is going to give you type B blood. Interestingly, if you have 1A and 1B, IA and IB, you're going to be type AB blood. And if you are totally recessive, if you have 2 lowercase i's, you're going to have type O blood. So these 3 alleles will give you 4 different types of blood, A, B, AB, and O. You guys are probably thinking about blood types and thinking about pluses and minuses on your blood too. That's a totally separate gene that sorts independently from ABO. So we won't make things more complicated. But the Rh factor, plus or minus, is a different gene. All right, the last complexity here is that some genes have multiple phenotypic effects, and the vocabulary term for this is pleiotropy. So pleiotropic alleles cause lots of things. So you have some hereditary diseases such as cystic fibrosis, sickle cell disease, where one mutation actually causes a whole host of problems. Those get more complicated to look at because the pleiotropic effects of the mutation end up making things confusing, masking what's actually going on. All right, so let's extend this even more. So if we add 2 or more genes, so now we're not talking about multiple alleles per se, we're talking about multiple genes, polygenic traits. The gene products might interact and create something different. A classic example of this is epistasis. So a gene at one locus will alter the phenotypic expression of a gene at a second locus. The classic epistasis example is Labrador retrievers. I have a yellow lab. So one gene determines the pigment color, whether your lab is black or brown. The other gene determines whether or not that color is actually deposited in the hair. So all yellow labs are either black or brown, but they are recessive for this allele, this gene that determines whether or not the pigment is deposited. So you can see this is going to look like a dihybrid cross, but you're going to have some that are black, some that are brown, and then some that could be black, and some that could be brown, but because of this E gene being recessive, they become yellow, and you get a completely different ratio, 9 to 3 to 4, black to brown to yellow. We'll work on one of these in class, but this is a way where 2 genes interact. And you can multiply this out, right? If you think about 2, 3, 4, 5, 6 genes interacting, you can start seeing where we get a lot of diversity in organisms and humans based on how things are interacting with each other. You can also have characteristics that are along a continuum. So this is now adding 2 or more genes for a single phenotype. They don't necessarily interact with each other, but they combine in different ways. Skin color is a classic polygenic characteristic. And so you can see here, if we think of 3 different genes controlling skin color, either dark or light for each one, you can go from a place where you're totally dark to a place where you're totally light, and you can end up all over the board for intermediate colors between dark and light. And even this is a simplification when I show you just 3 genes. Imagine if you add a lot of genes, this Punnett square is going to get bigger and bigger, and the range of outcomes is going to be greater and greater. Adding even more complexity, and I apologize for continuing to add it, but I run the risk as we show you genetics that you start thinking things are really simple and you can kind of determine by looking at 2 parents what their children are going to be. Environment also impacts all of this. We've already talked about epistasis a little bit. We've talked about how the environment can affect the DNA and the DNA replication. Characteristics that are influenced by both the environment and the genes themselves are called multifactorial. There's a lot of factors collectively influencing phenotype, and this causes outcomes that don't always line up with exactly what you think. The classic way to think about multifactorial characteristics is that you guys all know there are a few sets of identical twins at school. Identical twins very rarely look completely identical to each other, even though they are genetically identical, because even though they might live in the same home, even though they share the same environments, there are some environmental differences between the two that end up making for some differences, however slight, between identical twins. All right, so we have clearly moved into talking about humans, and that's a place that most guys end up with when they are thinking about genetics. How does this apply to you and to your family and different traits? Humans are not great subjects for genetic research. We do study humans. We try to understand what's going on with humans. The problem with using them in a research lab is that generation time is too long. It takes 20 to 30 years to get new offspring. Parents produce relatively few, unlike rabbits or flies or something like that. Parents usually only make 3 to 4, maybe 5 offspring. And breeding experiments are unacceptable, so it's completely unethical to just grab 2 people, make them mate to see what the offspring are like. That said, that is occurred in history. There are groups who have tried to do these breeding experiments, and they are uniformly decried as completely unacceptable. And even when they were done, they didn't produce any results that were worth doing. So people don't use humans to study genetics and patterns of inheritance, but we will use Mendellian genetics and our understanding of genetics to study human groups and try to understand what's happening with those groups. And the tool we use for studying groups of humans is called a pedigree. A pedigree is a family tree that just has genotypes on it. And let's show you one here. And what I like about pedigrees is that they basically are showing you a bunch of Punnett squares all at once. And they set up for interesting problems that you're able to solve. So the key on these, males are squares, females are circles. If males have the trait, they're colored in a color. If females have the trait, they're colored in a color. Anytime you see a line between people, they have mated or they have had children, and then you see children below them, firstborn on the left. So here are some grandparents. You see that grandpa had the trait for widow's peak. Grandma had the trait for widow's peak over here. 2 of their children had the trait. 2 of their children had the trait. These 2 people who both had a widow peak had a child. 1 of their children had a widow's peak. 1 of their children did not have a widow's peak. Each step in this represents a Punnett square, and so these are all things that can be sort of looked at and tried to figure out and do those test crosses to determine what her actual genotype is. Here's another one for tasting PTC, and you'll see a similar thing. PTC is totally recessive, but it does appear at different times in the generation. These are just close-ups of those. The thing with recessive alleles, and you've probably heard this language before, is that recessive traits only show up if someone is homozygous for the recessive trait. So there are heterozygous individuals who carry the recessive allele, and we'll call those carriers. There are some disorders where both parents can be carriers and not know it. They're both heterozygous. That means 1/4 of their children have a chance of having the trait. Most people who have recessive disorders are born to parents who are carriers of that disorder. So here's a family where dad was a carrier, mom was a carrier of albinism, and so 1/4 of the children will have an albinism phenotype. 50% of the children will be normal but be carriers, and 25% will be completely normal phenotypes and genotypes. Sickle cell is the same, and I feel like we've talked about sickle cell before, that homozygous sickle cell will only have sickled cells, but carriers will have sickle cell trait, and so they can pass this disease, sickle cell disease, onto their offspring while also having sickle cell trait. There are some disorders, and we'll wrap this up, and you guys can think about it some more, that are dominantly inherited. That means that if the dominant allele causes the disease, most, or at least 50% of the offspring will have that disease as well. They're pretty rare because they don't tend to have successful offspring, but dwarfism or achondroplasia is a form of dwarfism that's caused by this rare dominant allele. And so you can see that if you have a parent who has a dwarf phenotype, big D, little d, you have a mother who does not have a dwarf phenotype, little d, you're still going to see 50% of the children having a dwarf phenotype. Another type of dominant disease is Huntington's disease. And so Huntington's disease is a degenerative disease of the nervous system that can be dominantly passed down to children. Unfortunately, it doesn't have any phenotypic effects until 35 to 45 years of age, so most people who have it have successfully had children by that point. The problem with this is that they didn't know that they had a disease, and so this actually does get passed on a little bit more. And there's a lot of ethical issues around testing yourself for Huntington's disease, because if you test yourself and you are positive for Huntington's disease, there's not much that you can do about it medically. And then also your children know that they have at least a 50% chance of also inheriting that. So it becomes a very interesting ethical dilemma, as do many questions in genetics, which is why there are trained individuals, genetic counselors, who help people think about what's going on genetically and with their families and their children and things like that. And again, just to add that sort of clause again, that reminder, lots of things, heart disease, diabetes, alcoholism, mental illness, cancer, have genetic components, but they also have environmental components. So the interplay between genetics and environment continues to be important, and we continue to think about these things as multifactorial. There are lots of things going on. Genetics is only a small part of the picture for these. All right, that was a lot of stuff. That sort of flew through Chapter 11, but what I think you will see is that Chapter 11 is going to set you up for Chapter 12, and by the time we get through Chapter 11 and Chapter 12, you'll have played with some of these genetic problems, and things will make a lot more sense to you.